Hi, I'm Andrew Jackson, and I am going to be narrating this presentation on high-resolution passive profiling of contaminated sediments, design and application. This presentation is mainly focused on describing the abilities and attributes of the high-resolution sampler, as well as a how-to of its application, how to install them, and how to sample them. The results of the various field studies that have been conducted with the samplers will be presented in a different slideshow focusing on the outputs of the project. This project was performed by myself, Dr. Danny Rival, Dr. Paul Hatzinger, and Greg Livorno, and the funding was provided by the ESTCP program. The Sediment High Resolution Passive Profiler is a multifunctional device that is used to holistically look at the fate and transport of species in sediments. The device that's shown on this slide is really focused on the shallow sediment, 0 to 1 meter, and is meant to provide information at very high resolution, centimeter scale. It is able to be driven in by hand, but there are other versions of the device that can be used for deeper deployments or deployments where hand driving may not be possible, with a little bit different geometry, a little less resolution, but that are able to be driven in by either a geoprobe or a vibrocore device. And those devices can reach depths of 30 feet below ground surface and have been deployed to such depths in a variety of settings. The device is able to measure a number of parameters, and the first parameter are the dissolved pore water species with reasonable solubilities. And so those would be things like the geochemical indicators, the electron acceptors, nitrate, iron, sulfate, sulfide, methane, other dissolved gases, conservative transport tracers like chloride, and then contaminants that have a high enough solubility to be able to analyze without concentration. And those include things like the metals, chlorinated solvents, BTEX, energetics, explosives, all of which devices have been used to monitor. The equilibrium cells, as will be explained in the next coming slides, basically operate by equilibrating with the pore water and are diffusional samplers. For contaminants with much higher absorption parameters, very high KOW values, also referred to as the hydrophobic organic carbons. Those would include things like PEHs, PCBs, DDX, which typically must be concentrated in some form before they can be analyzed. The samplers can be adapted to use SPME fibers. SPME fibers are able to accumulate the contaminants and will eventually approach equilibrium with the pore water, but because of the sorptive nature of the fibers, accumulate the contaminant to the point where they can be readily analyzed. So the samplers are able to look at geochemical conditions. Those are parameters that enable you to evaluate how the geochemistry or biogeochemistry may be impacting the contaminant or availability in the case of metals. And they also allow you to look at the distribution of the contaminant within the sediment, whether that's the more soluble contaminants or the hydrophobic contaminants. But they also can be used to evaluate the velocity of the pore water, if the velocity of the pore water is large enough. And so there are velocity cells, and the velocity cells are on the back side of the sampler. There are 4 groups of these, and their function will be explained in a following slide. But the point of these is to be able to estimate the pore water velocity, which can be very important in understanding the long-term fate of contaminants or the flux of contaminants to a critical zone or receptor. The last function of the samplers is the microbiotraps. So in some cases, the microbial community can play an important role in the fate of contaminants. And so the devices are equipped with microbiotraps, and these are just cells in which a commercial product, bioset beads, can be placed. The bioset beads are able to accumulate microorganisms, and those beads can then be sent out for commercial molecular analysis to evaluate the community composition or even specific bacteria with specific functions like pH oxidizers or reductive dechlorination. The beads can also be used to accumulate the certain contaminants or compounds, and if appropriate, those beads can also be sent in order to look at the compound-specific isotope composition of the compounds in order to evaluate degradation. And that will also be explained in more detail in the following slides. So the sampler overall is about 34 inches. It has a slightly smaller zone of sampling. There are 3 sets of equilibrium cells, and every cell is capable of having a SPME fiber as well, depending on the resolution that the study requires. So this is on one side of the sampler. They're made of stainless steel or polycarbonate, depending on the design of the study and what species are of interest. On the back side of the sampler are the velocity cells, which measure velocity, and then 3 sets of microbiotraps. So to explain a little bit more how the devices are able to acquire samples in order to evaluate pore water constituents, this slide goes into more detail on 2 different aspects of the samplers: the equilibrium cells and the SPME fibers. So the equilibrium cells are essentially a reservoir of water that is separated from the pore water by a microporous membrane. This membrane can be made of a number of different materials: nylon, polysulfonone, stainless steel, depending on the application of interest. Dissolved species in the sediment pore water will diffuse across the membrane into the cell, and over time the concentration in the reservoir will match the concentration in the pore water. To evaluate the degree of equilibrium, the water is usually spiked initially with bromide solution, and the bromide loss from the cell can be used as a measure of the degree of equilibrium reached with the sediment pore water. And this technique is really useful for anything that doesn't sorb significantly to the membrane, and that can be measured with the volume of water that is available from the reservoirs. For the samplers in this study, those reservoirs are generally around 10 to 13 mils of water, and so that volume of water can be used for whatever analysis is of interest. Typically, the water is used to look at multiple species simultaneously. So that would be chloride, the major electron acceptors, dissolved gases, metals, and in some cases dissolved organic carbon. If a particular parameter requires more volume than is provided by a single cell, then cells can be used for various purposes. So for instance, the first cell might be used for the contaminant concentrations and the second cell for geochemistry or metals. The reservoirs typically require 21 to 28 days for full equilibrium. And the reservoirs, again, are the cells pictured on this side of the sampler and are really just a cut out into the material that is separated from the sediment by a membrane, as pictured here. For hydrophobic compounds, in which usually the detection limit from the volume of water provided is not low enough, the SPME fibers are included in the devices. So the SPME fibers are a inert core with a highly sorptive material coated on it. The fibers are placed on top of the equilibrium cells, exposed directly to the sediment, and contaminants, particularly hydrophobic contaminants, will diffuse into the sorptive layer on the fibers and will accumulate until an equilibrium is reached between the concentration in the fiber and the concentration in the pore water, which is based on the hydrophobicity of the compound. The fibers are generally pre-equilibrated with a performance reference compound, and the performance reference compound loss can be used to evaluate the degree of equilibrium reached. And there are many more details on SPME sampling for sediments in other publications or presentations. This is an existing technique that has been well demonstrated and essentially been adapted to be able to apply the SPME fibers in a horizontal as opposed to vertical orientation, which allows you to get more SPME length per depth, which gives a lower detection limit and allows a higher resolution of sample analysis. Pore velocity can be estimated with a technique that was developed which allows the mass transfer of a conservative species to be related to the velocity outside of the velocity cells. So the velocity cells, as mentioned, there are 4 sets of these on the sampler that cover the overall length of the sampler. These cells have different specific surface areas. So there's a close-up here, and the opening of the cells is different, but the volume of the cells is the same. This allows the loss of bromide to be varied for a single time point of deployment. So for a 21-day deployment, the cell with the largest surface area is going to equilibrate more than the smallest. And this variation allows a curve to be developed which describes the loss of bromide out of the cells for a single time point with respect to the specific surface area. And that loss is governed by the specific surface area, the A to V term, the time deployed, and a mass transfer coefficient. So with a single deployment, we can have 4 data points based on the area to volume ratio, and we can then estimate the mass transfer coefficient. The mass transfer coefficient of the bromide out of the cell to the pore water is also dependent upon the velocity outside the cell. And that relationship, which was developed in a paper by Schneider et al. And is available, shows that as the velocity of the pore water increases, the mass transfer coefficient increases in a nonlinear manner, and therefore if the geometry of the cell and the thickness of the membrane is known, then the mass transfer coefficient measured by the deployment can be evaluated with respect to the velocity outside the cell. Now, it should be noted that the resolution at very low velocity is not very good. So for velocities less than about 3 centimeters per day, it's very difficult to determine the true velocity or a velocity that's different than 0. But for velocities over 3 centimeters a day, the resolution is adequate, and for situations in which there's upwelling or tidal influences, the samplers can provide valuable information on the rate of exchange that could be occurring. And for more details on the specifics of the measurement technique, I refer you again to the published paper in groundwater. So for measurement of microbial community and compound-specific isotope analysis, it's a technique which was adapted from groundwater by a company called Microbial Insights, which developed a proprietary sorbent, bioset beads, which are capable of accumulating organisms and accumulating some types of contaminants, the most common being chlorinated solvents, although there are other contaminants that would be amendable, things like BTEX. And those bioset beads can be placed in these slots located on the back side of the sampler and are separated from the sediment with a stainless steel screen to allow rapid flux between the pore water and the beads to promote bacterial attachment and contaminant absorption. The bioset beads are basically equilibrated with the sediment again for 21 to 28 days, and then are sampled, which will be described later. And the beads can then be shipped to Microbial Insights for commercial analysis, molecular analysis for a wide variety of bacteria, or for CSIA C13 analysis of the absorbed contaminants if appropriate. So the beads are deployed in the samplers, the microbes attach to the beads, and then the beads are sent for qPCR for microbial population or key functional genes. And this has been commonly used for chlorinated solvents, particularly in groundwater, to evaluate whether or not specific reductive dechlorination is occurring, to look and see if the biogeochemistry is appropriate for particular types of degradative processes, or if co-metabolic processes are occurring. It can also be of interest to other studies depending on the contaminant and the conditions. The beads, as mentioned, can also be used to evaluate the isotopic composition of appropriate contaminants. So the contaminants absorbed the beads, and then the beads are sent for CSIA. In the most common occurrences, C13 is analyzed of chlorinated solvents to understand if the parent compounds have been fractionated or daughter compounds have been fractionated. And that usually includes TCE and some BTEX compounds as well. So the focus of this presentation is really less on the abilities of the sampler, which were just quickly reviewed, and more on how to actually use the samplers. So their preparation, their assembly, and how they're sampled. And the remaining slides are going to go through in detail on those areas. And to support this discussion and these slides, I want to draw your attention to a video that is available on YouTube, which demonstrates the assembly of the samplers, which can be used as a reference for anyone that would like to utilize these and supports this presentation with more detail. So preparation and assembly. The first thing that should be thought about is material compatibility. The samplers are available in stainless steel and polycarbonate. It's also possible to make these out of other materials, but those are the two probably most useful materials. Stainless steel is excellent for almost any constituent monitoring except for metals. The stainless will leach metals and it will have an impact. Polycarbonate can be used for most soluble species. It would not be appropriate for hydrophobic compounds. Also, for evaluation of metals, you should avoid the use of stainless meshes. Nylon meshes are available. And so throughout the presentation, if you see stainless meshes but are interested in heavy metal or metal monitoring, you can simply substitute those stainless meshes with nylon. Preparation. The samplers can be prepared prior to installation, although they shouldn't be stored for extensive periods. They can also be prepared on site as needed. The samplers should be stored in the bromide solution in which they're prepared so that the bromide does not change from the initial preparation. And if oxygen is concerned, as it is sometimes in metal sampling, the water can be degassed using a nitrogen purge, or the samplers can be prepared and then the samplers degassed after they're prepared. Long-term storage of the HRIP using SPME fibers should be avoided. The SPME fibers will slowly leach the performance reference compound depending on the contaminant of interest. That can be faster or slower. And so over long periods, the fiber's initial concentration will change. And so it's preferred if the samplers are prepared within 24 hours of their application. In general, depending on what type of contaminant is of interest, if hydrophobic organic compounds are being evaluated, the overall assembly would be to preload the SPME fibers prior to deployment with the appropriate PRC. This can take weeks in some cases, or acquire them from an appropriate source. You would assemble the microbial trap compartments first, submerge the sampler in a bromide solution, assemble the remaining sampler, which will be detailed in the following slides, and then store the submerged sampler in bromide solution until installation. And the construction and assembly of these samplers will be detailed in the following slides. So if the focus of the study is to evaluate hydrophobic organic compounds, which will include the use of SPME fibers, then this slide will detail the assembly of the equilibrium cells to allow that. So the top picture is one of the samplers with all of the membranes, cover plates removed. It's simply a stainless steel body that cells have been milled into, which can be filled with water and covered in order to allow monitoring of dissolved pore water. The materials that need to be assembled include a Viton gasket, pictured here, a membrane of appropriate material. In this case, I'm showing a 0.45 micron nylon membrane, a stainless steel mesh with the SPME fibers attached, another stainless steel mesh to protect the SPME fibers from the sediment, and a top cover plate to hold all of the previous materials in place. The SPME fibers can be attached to the stainless steel mesh using silicone, a syringe, and then the SPME fiber. So to look at this in a little bit more detail, the preparation of the SPME fibers, the syringe is filled with silicone. The fibers are then held using gloves, and a very small droplet of silicone is applied to the end of each SPME fiber, which can then be pressed onto the stainless steel mesh in order to affix it to the stainless steel. This is pictured with the cover plate on. This is important so that the SPME fibers will line up appropriately with the openings of the top cover plate during final assembly. So after all the fibers have been attached, the cover plate can be removed and the stainless steel mesh can then be applied to the sampler. So to look at the assembly in more detail, the first picture shows the sampler. It shows the Viton gasket, the membrane, the stainless steel with the SPME fibers attached, the top stainless steel, and then the cover plate. So the first step would be to submerge the whole high-resolution passive profiler in the bromide solution. Usually the bromide solution ranges from 100 to 300 milligrams per liter. The exact concentration is not really important as long as the initial concentration of the bromide solution is known. In seawater situations, higher bromide concentrations can be used. In freshwater, 100 to 300 is generally the upper range to prevent density gradients from occurring. The first step is to remove trapped air in the screw holes. This is usually accomplished by taking a syringe with water in it and injecting water in all of the screw holes to remove any air bubbles that might get trapped under the membrane. And the video that was mentioned in the previous slide demonstrates that for you. The next step is to place the materials. So in slide A, it shows the sampler with none of the materials. In slide B, you can see that the gasket, the Viton gasket, has been placed over the cells. In picture C, the membrane has been placed over the cells. In slide D, the stainless steel mesh with the fibers attached has been placed. In slide E, the top stainless steel mesh covers the fibers to protect them. And in slide F, the cover plate has been placed. And in slide G, the cover plate has been screwed to the sampler using stainless steel screws. And again, all of this is demonstrated in the accompanying video on YouTube. In cases where hydrophobic organic compounds are not involved in the study, and only species that can be directly measured in the equilibrium cells, the cells are assembled identically to before, but we just leave off the SPME fibers. So in this case, again, we have the equilibrium cells in which the cells are filled with bromide solution. We have the materials, the Viton gasket, the nylon membrane, the stainless steel mesh, and a cover plate. With the difference being that there's only one stainless steel mesh, because the second stainless steel mesh with the SPME fibers is not required for this type of study. So as before, the first step is to submerge the whole sampler into a bromide solution. Air is then removed from the screw holes using a syringe and needle by injecting water into each hole. After that, the Viton is placed over the cells. Then the nylon membrane is placed over the cells, the stainless steel mesh, and finally the cover plate. This is then affixed to the sampler using stainless steel screws, and this is all demonstrated in the YouTube video which was mentioned earlier. In order to prepare the remaining cells, which are on the back side of the sampler, and we're going to start with the velocity cells. The sampler is again submerged under the bromide solution. In this case, the equilibrium cells have already been prepared. The sampler then is turned over so that the equilibrium cells are facing down and the velocity cells and microbial traps are facing up. The velocity cells are submerged, and materials used to assemble are a Viton gasket, a stainless steel lower cover plate. This cover plate basically allows the size of the opening to be regulated, a larger gasket that goes over the stainless steel plate, the nylon membrane, a nylon mesh, a stainless steel membrane, and a stainless steel cover plate. The nylon membrane is again in this case a 0.45 micron. And for velocity measurements, if the relationship that was developed in the Schneider et al. Paper is to be used, it is important to use the same nylon membrane, the same nylon mesh, and a stainless steel mesh so that the total thickness between the water in the velocity cell and the sediment remains the same. In order to assemble the cells, the sampler is submerged. We remove all the air bubbles using a syringe and needle as before from the screw holes. And the reason this is important and it's being emphasized is that if you don't remove the air bubbles, after assembly, air bubbles can work their way up under the membrane, which reduces the specific surface area that's available for contact. The next step is to add the lower Viton gasket. The next step is to place the lower stainless steel plate and be sure to ensure that the size of the holes lines up with the Viton gasket. Then a upper Viton gasket is placed over the lower stainless steel plate. Again, you can see the holes. a nylon membrane is placed over the gasket. The nylon mesh and stainless steel screen are placed over the membrane. A cover plate is placed, and the cover plate is screwed down using stainless steel screws. And this picture also shows you the sampler with the biosep beads placed in the microbial trap and the velocity cells completely assembled. So the assembly of the microbial traps. The microbial traps were assembled prior to placing the HRIP in the bromide solution. So before the sampler is submerged, we assemble the microbial traps. And the reason for this is very simple. The biosep beads float. And if you submerge the sampler, it is very difficult to get the beads to stay in the cell in order to be able to affix the stainless steel screen. So before submersion, the biosep beads are going to be added to the cells. There's a stainless steel screen and a cover plate. The biosep beads are added to the microbiotrap cells shown above here. The stainless steel screen is placed over the microbiotraps, and the cover plate is then placed over the stainless steel screen and affixed with screws. The next part of this presentation covers the installation of the samplers once assembled. And as mentioned, the samplers are generally driven in by hand. They can be driven in by hammer, shown here. They can be driven in with a fence post driver. In some cases, they can just be pushed in if the sediment is soft enough. If required, they can also be inserted using more mechanical devices such as a vibrocore geoprobe. But in this presentation, we're primarily going to focus on the insertion by hand. You can also see that it's possible to deploy these from a boat by wading in from shore, and they can be deployed in open water, in marshes, in bays, estuaries, numerous applications of almost any type of sediment. The only problem that can occur is if there are cobbles or gravel, which make the insertion of the samplers too difficult. And in this picture, just showing you a couple of real-world applications. This is a stainless steel sampler that was equipped with SPME fibers that is being placed in Abrams Creek at Quantico to evaluate PCBs. This is a sampler being driven in to the embayment at Quantico to evaluate DDX. This is a polycarbonate sampler, and on the back you can see the biosep beads that are present. That was driven in to Canal Creek at Aberdeen Proving Ground, shown here, to evaluate heavy metals. Transporting to a field site. The samplers can be deployed off-site in a lab or even a hotel room, and they can then be transported to the field. A couple of issues that should be thought about. One, the samplers need to be stay submerged. They can be out of the bromide solution for hours without drying out, but in general it's best to keep them submerged. Plastic containers can be used to transport them. They can then be transferred from a truck or a van to a boat, and then a separate container used to keep them submerged until they're ready to be deployed. In the case of heavy metal sampling, if oxygen is a concern, a sealed container should be used. Often we use a PVC pipe that's been threaded to allow end caps. The water is deaerated, the sampler is put into the pipe, the pipe is capped, and that allows transport without oxygenation. The pictures here just show the samplers being transported to the field in a boat, and the samplers are stable for fairly long periods of time if kept submerged. So as mentioned, installation can be done from shore or by wading. In the case of wading, the consistency of the bottom sediment is important. In very soft sediment, this can be very difficult. In marshes, boards can be placed to prevent the damage to the marsh and also to allow a stable platform on which to drive the samplers. In this case, the sampler is being pushed in by hand due to the soft nature of the sediment. And then, if required, a hammer can be used to further drive the device in. The rope is going to be used to attach the sampler to a buoy or a stake to ensure that the sampler can be found after the 3 to 4 week deployment period. In this case, the bottom was sufficiently solid to be able to allow us to wade out, and because of that, we used a fence post driver to drive the sampler into the sediment. Samplers are generally pretty immune from damage from being driven in. In the case of polycarb, it's recommended to place a board between the sampler and the hammerhead so that the polycarbonate does not get damaged. Installation from a boat is also possible. So again, this is Canal Creek at Aberdeen Proving Ground. The samplers have been attached to a drive rod to allow us to be able to drive this in the bottom of the canal. The rope is tied to it to be able to affix a buoy in order to be able to retrieve it at a later date. And here you can see the hammer being used to drive the sampler into the sediment. Here the sampler is being driven in using a sledgehammer into marsh from the boat. And here we've actually attached 2 boats together to make the platform more stable. So almost any kind of arrangement can be adapted. Larger boats are obviously easier, they're more stable, but usually the sediment is soft enough, even in the case of sand caps, that the samplers can be fairly quickly and easily driven in without much of an issue. It is important to mark the samplers. Oftentimes changes can occur in the sample location due to the water rising to make it difficult to find the samplers. In open water, we generally use buoys, and the buoys are attached to the samplers using rope. This is pictured here. You can see the sampler that's been deployed into a shallow bay attached to a buoy. In this case, there's a sampler driven into the marsh that's then got a rope that's attached to a stake. And then in this case, again, you can see samplers deployed to a shallow bay, and we've marked the location with a buoy. It should be kept in mind that if there's significant wave action, large buoys can dislodge the samplers. Usually in the case of the stainless steel samplers or polycarbonate samplers, they're driven deep enough where that's never been an issue, but in some extreme cases it is possible. If that's of a concern, then instead of using buoy, it's possible to use an underground cable that can then be retrieved using a drag line. Retrieving the samples. Again, the samplers can be retrieved either by boat or by wading. Generally, they're removed by either pulling on the attached rope or by grabbing the sampler with your hand and pulling. Because of the length of the samplers, there is a suction that can occur, and it can be difficult to pull them. In that case, we recommend that a sharpshooter shovel or a breaker par can be used to loosen the sampler. And generally what we suggest is that the breaker bar shovel is put along the short axis, and then the sampler can be slowly wedged back and forth while someone else pulls, which allows the vacuum to be released and the sampler to be removed. So here's a picture of a sampler being removed. It's now come out of the water. It's then placed in a plastic bin, and it's recommended that the sampler not be cleaned. So leave the mud and sediment on the sampler until the sampler is ready to be sampled for all the different compartments. This can be done right on the boat. It's a little bit more difficult. Usually some fuel station, a parking lot, any place that a table can be set up nearby is deployed, which allows the sampler to be sampled a little bit more easily. If you wish, the picture in the lower left can be clicked on in order to be able to show a short video of the samplers being removed. Field sampling and analysis. The samplers should be sampled as soon as possible after they're removed from the sediment to prevent any type of changes, which are typically due to redox changes in the geochemical species. We often use a van, which can be set up, driven into the site. Tables can be deployed in the van. This allows protection from the weather. If it's cold, the van can be heated. And if it's raining, obviously that's a benefit. In this case, we've erected tents, and the tables are being placed under the tents. The sampling is going to be covered in the next slide. This is just basically an overview of a field deployment. You can see the sample bottles have been pre-positioned to allow rapid sampling. There is a screw gun to be able to remove the cover plates if required. Syringes are available for each sample cell, and with 2 people it normally takes about 30 minutes to sample the equilibrium cells and 45 minutes to sample the sampler, including the SPME fibers, equilibrium cells, biosep beads, and velocity cells. Some parameters need to be sampled on site. In this picture you can see a very small device, which is a portable spectrophotometer, which is used to measure dissolved iron and sulfide, which is highly reactive and usually measured in the field. So a little bit more detail on how the different types of cells are sampled. The first step is to remove the excess sediment over the membrane, usually by scraping with a plastic tool. And so each cell, the sediment is removed right before it is sampled. And again, this protects the cell, mitigates the transfer of oxygen, reduces the changes in the pore water during sampling. After the sediment's been removed, the surface is blotted dry with a chem wipe to remove any surface water over the cell. A needle is then placed just through the membrane. This needle is used to remove the vacuum as the water is removed. Then a syringe and a needle is inserted into the cell, and the water slowly removed by pulling on the syringe. In cases where there's issues with sorption, glass syringes can be used. If you're only interested in geochemistry, plastic syringes are much easier to use. The water from the syringe is then distributed into the appropriate sample containers depending on the analyte of interest.
I'm taking a video just to show you how we do this. So we have to, it's part of one of our deliverables.
Okay, for the velocity cells, the exact same technique is used. The sediment is removed from the screen by scraping gently. A chem wipe then is used to absorb the excess surface water. A needle is placed through the screen to right below the membrane. A separate syringe and needle is then placed through the membrane. The water is removed and the sample distributed into the appropriate container.
The wall.
Okay, ready?
Once the equilibrium cells have been sampled, in cases where SPME fibers have been deployed, the SPME fibers are then sampled next. And to sample these SPME fibers, the first step is to remove the stainless steel cover plate. So the screws are removed. Once the screws are removed, the plate is removed. The top screen is removed, and the screen with the SPME fibers is gently removed and pasted on a clean surface. Generally, we use aluminum foil. After the screen's been removed, using a ceramic cutter, the fibers are cut adjacent to where they were affixed to the screen with the silicone. The fibers are then placed on a clean surface, gently wiped with a chem wipe. The fibers are cut into appropriate lengths, usually 2 centimeter pieces, and then the fibers placed in a sample container, which also contains an appropriate solvent to prevent loss of the contaminant during shipping. The top picture can be clicked on for a short video showing the removal of the plate and the sampling of the fibers.
Great. Thanks.
In order to sample the bioset beads, the cover plate is removed, the stainless steel screen covering the bioset beads is removed. The fibers are removed using a spatula and placed in a sterile stainless steel screen. The screen can be cleaned between sample cells using ethanol. The beads are then gently rinsed using sterile phosphate buffered solution, using a large syringe with a needle, and the solution is gently squirted on the beads to remove any loose sediment or sand, so that the beads can then be placed in a sterile plastic container for shipment on ice for molecular analysis. So the preceding slides covered the assembly, deployment, and sampling of the samplers, the HRPP samplers. As again, there is more detail in the YouTube video. And while this presentation is not designed to cover the demonstrations and the results that were obtained from the various deployments, I did want to at least give you a short preview of the type of data that is collected by these samplers. Examples of the HRPP deployments that have been done to date were for PAH monitoring, PCB monitoring, DDX, chlorinated solvents, and metals. They have been deployed at water depths from 0 to 4 meters. They've evaluated the geochemistry, conservative tracers, pore velocity, microbial communities, and CSIA. And the graph on the right is just one example of the type of data that can be obtained. So this is a depth profile, centimeters below ground surface, for a marsh, in which you can see the chloride concentration increasing with depth. Chloride is often used as a conservative tracer for transport. You can see the geochemical indicators: nitrate, sulfide, iron, and sulfate. We see a little nitrate at the surface. It completely disappears fairly quickly. Iron concentrations are generally low throughout. There are some peak sulfide concentrations and a very nice sulfate profile, which decreases with depth. Metals we evaluated here were arsenic, zinc, and mercury. You can see that the zinc profile decreases proportionally with the sulfate profile, probably due to sequestration with sulfide. Arsenic increases with depth, and mercury was generally low throughout the profile. Lead was highest actually in the surface water and decreased with depth, and PCBs also decreased with depth. So again, there's much more detail on the results of these deployments for each of the deployment sites available in other presentations and publications, but the samplers do have a wide utility and can provide very high resolution data, which is conducive to modeling studies and for understanding the fate and transport and long-term fate of the contaminants in a variety of sites. Finally, I'd like to end with my contact information. If anybody would like to discuss further the application, uses, assembly, sampling, or any other details of the devices, I'm happy to help with that. You can contact me at Texas Tech University and the Department of Civil, Environmental, and Construction Engineering. My phone number and email address are on the slide, and I look forward to working with anyone that would like to utilize the device or just to understand better how it might be used to help your specific site. Thank you very much, and I hope this was helpful.